So today I'm going to be talking about transcription and translation and gene expression. So first, gene expression is the process by which information stored in a gene is turned into a functional protein product, which determines observable traits of an organism. So first, transcription. So in transcription, an RNA copy of a DNA gene is synthesized. So these two blue strands and the nucleotides coming off of them make up a DNA gene, and this little gray tannish circle, it represents the RNA polymerase, the enzyme responsible for transcription. So an important part of DNA is a nucleotide, and DNA is made up of many little nucleotides to make each strand. So the RNA bases pair to the DNA bases because of complementary base pairing, where only some bases match up with others. And the process of transcription happens in the nucleus of the cell. Additionally, hydrogen bonds between the bases of the two separate DNA strands keeps them in their double helix. Hydrogen bonds are also formed between the RNA bases and DNA bases through complementary base pairing. And covalent bonds link the nucleotides together in the DNA strands. Okay, so next is translation, which is the synthesis of polypeptides from mRNA. So we're going to start with a large ribosomal subunit. And then we add the small ribosomal subunit. And so each subunit is made from rRNA and proteins. And so together they make up the ribosome where translation happens. And so the mRNA travels to the ribosome to synthesize polypeptides, and it kind of attaches to translate. And a sequence of 3 nitrogenous bases on the mRNA make up a codon. Each codon codes for a specific amino acid to be added to the polypeptide, and the codons together makes up the genetic code, which is the rules living organisms use to translate information encoded in genetic material into proteins. So amino acids are carried to ribosomes by transfer RNA or tRNA showed here. And each tRNA has an amino acid towards the top, as shown here. Each tRNA also has an anticodon at the base, which pairs to a codon on the mRNA through complementary base pairing. And this just shows the process where the tRNA brings the amino acid and meets up within a codon on the mRNA to form a polypeptide chain. And here you can see the tRNA doing that. And finally, protein folding occurs where there are covalent bonds between the amino acids, and these amino acids fold and kind of twist up into a protein, which is our final product there. And now the process of gene expression is done through transcription and translation. Thank you for watching.